All right. And our next speaker is Dr. Shingo Kajimura, professor of medicine at Harvard Medical School and BI Deaconess Medical Center, and an investigator of the Howard Hughes Medical Institute. Shingo's laboratory has pioneered our understanding of adipose tissue biology, thermogenesis, and energy homeostasis, uncovering molecular mechanisms that regulate whole body metabolism. Today, he will present molecular basis for fuel selection. Please join me in welcoming Shingo to the stage.
Well, thank you so much for having me, and thank you for the opportunity, and really great to be here. It's always great to sort of catch up with friends and colleagues, and it's beautiful. Thank you. So Marsha gave an amazing talk on mitochondria metabolism, and I think it's a great follow-up for me to talk about sort of fuel selection, what determines how we in cells decide what fuel to use. And the idea is that, let's say, where can I do this? There you go. We have a fuel preference, right? I mean, sometimes you want to eat meat, salad, maybe carb, or maybe sugar. And then this sort of change, the preference I would call, is change depends on the environment and adaptation. For example, if you go start running, what happens is muscles start to use glucose, right? It's the favorite substrate, and then it lasts for probably a few minutes to an hour. And then eventually they switch to fatty acid. And if you do this in a starved condition, they start to proteolysis, muscles start to provide amino acid, they start to use something else. Same thing if you go to outside in winter in Boston, and then first thing is that you shiver. Second, brown adipose tissues start to generate heat, and then the brown fat love glucose, right? That's how FDG PET scan work. And then, but it doesn't actually, brown fat uptake a lot of glucose for the probably first half an hour or so, but eventually they switch to fatty acid. It's called fuel switching. And this fuel switching idea is often discussed in the context of fuel availability, right? In other words, peripheral tissue have to use something else and save glucose for the brain, right? That's sort of the idea of fuel selection, but switching. But I'm not sure that's really the whole story, because the fact is your glucose level remains pretty much the same even after you run full marathon or you stay in the cold for a few days or even. So that it has to be, this fuel selection and switching has to be matched with the internal changes. Okay. Another one is that, for example, there's also a big change in the metabolic disease, particularly for type 2 diabetes, obesity, or many of you know the glutamine addiction in certain cancers, right? Why certain cancer have to use glutamine? What is the molecular basis of glutamine addiction? So we really want to understand the molecular basis for fuel choice. And one of the bias, hypothesis or vision, we will say, is that maybe one of the major decision is at the level of mitochondria, specifically mitochondrial inner membrane metabolite transport. As you know, in large, we have three sources. One is glucose, pyruvate. Second is fatty acid, and third is amino acid. And then, but the point is that mitochondrial inner membrane is essentially impermeable to metabolites, a very tight membrane. So this means that every metabolites that go in or out require designated transporters, okay, the cardioproteins. But still, 2026, many transporters are, as we called, orphan carriers. We don't know the substrate. We don't know the function. There are a lot of things that we still don't know. It's amazing to see that. And so why this? Why so hard? My view is there are two reasons, mostly technical reasons, technical challenge. One is the computational prediction of the substrate still is not that accurate. One of the great example from our view, for example, the brown fat, I mean, we've been studying for many years, is that probably the most well-known protein is called uncoupling protein 1, UCP1. This is one of the mitochondrial inner membrane protein. Another one's called SLC25A7. I'm going to talk more about SLC25. SLC25 is largely, mostly in mitochondrial inner membrane protein. And then this is one of those. When about 35 years ago, UCP1 was cloned, and then soon after, a lot of people claim, oh, I found UCP2, UCP3, 4, 5, 6, 7. And assuming that they're all uncoupling protein means that they transport protons. Later, people figured out that actually only UCP1 does transport protons, and the rest, they don't do that, even though structurally very similar. Another one, even more, the UCP1 in mammals is a protein carrier. It transports proton and it's thermogenic, mitochondrial thermogenic protein. UCP1 gene exists in fish and reptiles, birds, marsupials, but they're not proton carriers, even though structurally very, very similar. Computationally so far, we cannot predict that. We still don't know what they're doing. But point is, the computational prediction, maybe in the future is better, but we can't really predict the substrate accurately. The second issue of doing this is that reconstitution of mitochondrial membranes is notoriously difficult. Making recombinant proteins are hard. Many of them are not properly expressed in E. coli, so that we really have to do something else. And then for the last 15 years, my lab been studying brown fat as a model of thermogenesis, a model of mitochondrial biogenesis. But at one point, I realized that this can be a great tool to deophanize some of the unknown mitochondrial carrier proteins, because brown fat is brown because of mitochondria, right? Mitochondria has a lot of ions. Brown fat contains mitochondria, probably the highest density in mammalian species, so that you can get a lot of mitochondria. So I thought maybe this is a great tool to purify mitochondria. The analogy I use is that, okay, we all use 293 cells, HEC293 cells for transfection, CRISPR screen, all kinds, because it's easy to grow, easy to transfect, but most of us actually don't study using HEC293 cells for kidney biology, but they are kidney cells, right? Kidney epithelial cells. So the same analogy, maybe instead of studying biology of brown fat, I thought maybe we can use this as simply a tool to purify mitochondria, use as a bioreactor to produce mitochondrial inner membrane proteins. We can study sort of reconstitution rates. For example, we now can do this in a liposome system. So this allows us to reconstitute and study flux, transport activities, all kinds. So one of the earlier success we did was using brown fat as a tool was to discovery of the mitochondrial BCA carrier. For example, branchy chain amino acid. So you'll notice an essential amino acid means that every single cell needs to use BCA for protein synthesis. Okay, anabolic reaction occurs in every single cell, which occurs in the cytosol. BCA get into the cytosol, activate mTOR, particularly leucine, induces protein synthesis. Some of the cells, very metabolic active tissues like brown fat, for example, bring BCA into the mitochondria because the first deamination enzyme, BCAT2, exists in the mitochondria, means that brown fat have to bring BCA into the mitochondria to initiate catabolic reaction. So that we use brown fat as a discovery tool, led to this sort of identification of SLC25A44. It's a terrible name for mitochondrial BCA carrier, and then we're able to reconstitute this and then we do a lot of mouse models now. So this is one example. Now, and then a couple more years now, then still hard, I have to say, the one protein takes probably, 6-7 years to deophanize and study the biology, but now we identify some of them, SLC25, 34, 35. It's a carrier for phosphoenolpyruvate. 48 is choline carrier. And then the one that I'm going to tell you today is about this another one called SLC2545, which is for trimethyl lysine. I'm sorry, this is a terrible name, but again, the deorphanizing, identifying substrate itself, it's important, but it's not the goal for us. The goal for us is to ask biology or pathology, right? And the identification of those molecules allow us to manipulate the pathways, let's say overexpression or knockout, then it's allowed to study biology. And hopefully even bigger picture is this will take us to the direction that we never thought about. And then I think this is a great example. I'm going to tell you more sort of unexpected area that I've been studying. So what is trimethyl lysine? Honestly, I didn't know anything about trimethyl lysine until we studied this. Trimethyl lysine is a methylated lysine. It's a trimethyl moiety here. It's a lysine residue. And then this, I didn't know again, it's being known, this trimethyl lysine, getting to the mitochondria and then hydroxylated and in the mitochondria spit out and eventually converted to one called gamma beta or butane. And even this is a precursor for L-carnitine. Then you probably heard about carnitine. I heard about carnitine. Carnitine, same again, hazardous of trimethyl moiety, is pretty important molecule for fatty acid oxidation. Marsha talked about a little bit about this. So the carnitine is a key metabolite to conjugate to make acyl-carnitine. Long-chain fatty acid can't get into mitochondria, right? You have to conjugate with carnitine. That's how you bring those long-chain fatty acids into the mitochondria through CPT1, CPT2. It's called carnitine shuttle, right? So then that's where we can bring those particularly long-chain fatty acids. And then it's interesting, carnitine, mostly we take carnitine from food. Food is the major source. Over close to 75% of total carnitine in our body come from food and 25% come from de novo synthesis. That's the pathway, de novo carnitine synthesis. If you eat meat, okay? If you happen to be vegan, strict vegetarians, or herbivores, 95% come from de novo synthesis. So we have a way to adjust carnitine synthesis, depends on what kind of food you eat. Okay, I'm going to get to the point. Now, then the point I want to say is biology. So just briefly, we're able to reconstitute this protein in the bacterial system. Now we do in the liposome is that this protein transport TML, trimethyl lysine. We did some structural study through the collaboration with Liz and Sun's lab. We identified some key substrates. We can mutate. We do that. And then I also want to emphasize that the two groups that around the same time essentially reach the same conclusion, so that it is gratifying for us to see that three independent groups reach the same conclusion that this is a carrier protein for trimethyl lysine. We made knockout mouse. And then, by the way, this molecule is highly expressed in the liver and the kidney. That's where de novo synthesis of carnitine occurs. And then we can do tracer experiments. For example, we can put D9 TML, and then we can see mouse and the control mice or cells, hepatocytes can make those metabolites, particularly called L-carnitine, but knockout mouse can't make that. So unexpectedly, we generated a mouse model that can't make carnitine, sort of carnitine deficiency in that sense. And so this actually takes us to the direction I never thought about. Oh, by the way, those two papers, mostly the culture cells, but also they found the same thing. Then we look into in vivo physiology. And then first we found this knockout mouse has some low carnitine levels. Then we measure whole body energy, sort of metabolism, energy expenditure. This is a way to measure this. They're using metabolic cages. You throw a mouse in a chamber, you measure oxygen consumption rate, CO2 release, and then to see how, for example, this is called RER, respiratory exchange ratio. That's the way to test assess the fuel selection within a whole body level. For example, if a body primarily use glucose or carbohydrate, then you consume one molecule of oxygen and release one molecule of CO2, right? So that was stoichiometry, ratio of VO to CO2 per VO2 is 1. So that if you calculate RER, respiratory exchange ratio, body use strictly carbohydrate, this become 1. If body use fatty acid, this value goes down because they clearly consume more oxygen and then releasing CO2. So 0.7. We noticed that the knockout mouse, carnitine deficient mouse, tend to show higher RER, suggesting that they prefer to use carbohydrate over fatty acid compared to control mice. When you purify mitochondria from, let's say, heart or liver, give palmitate as a substrate and measure oxygen consumption rate, knockout mouse can't use this as a substrate. Oxygen consumption levels lower. If you give pyruvate as a substrate, alternative substrate, they're perfectly fine. So somehow knockout mouse can't use fatty acid well. One robust way to induce fuel switching in the whole body level is cold exposure. As I told you earlier, if you throw mouse or you are exposed outside, and then first they start to use glucose for the first minute to up to an hour, but they have to switch to fatty acid. If you, let's say this is a core body temperature, rectal body temperature mice, control versus knockout, and then you expose these mice in a 6-degree temperature, knockout mice are able to maintain the body temperature up to 2 to 3 hours. Then after 4 to 5 hours later, they develop hypothermia, means the body temperature drop below 27 degrees. Now, if you give carnitine, supplement carnitine to knockout mouse, you can rescue it. So this means that the hypothermic phenotype of knockout mouse is due to carnitine, defect in carnitine synthesis. When you look at the serum levels, like for serum lipidomics, control mouse can make a lot of acylcarnitines. Okay, and this is a way to bring long-chain fatty acid into the mitochondria so that not control mouse will make a lot of acylcarnitines, but knockout mouse can't do that, right? Because of the lack of carnitine. So far what I told you is that we happen to identify this transporter without any expectation that it be important for the synthesis, de novo synthesis of L-carnitine. And if you can make carnitine is important for fatty acid oxidation. So if you knock out this pathway, body can't make enough carnitine, and they fail to adapt to cold. They can make acylcarnitine such that the brown fat thermogenesis is down. Then we ask, what else? Like, what else? I mean, I'm not going to talk about the data, but the fasting is another sort of way to induce fast fuel switching to fatty acid. But the one that I really never thought about is the response to GLP-1, okay, the weight loss effect. GLP-1 is sort of pharmacological way to induce fasting, right? That's how you lose weight. And then this is, I think GLP-1, everyone knows this, it's really completely transformed the obesity therapy for the last 5 years or so. And the primary phenotype is the mechanism losing weight is inhibit food intake, suppress food intake, right? Now, over 20 million people are taking GLP-1. 20 million people, probably amazing. But the response is highly, highly variable, depends on the person. Some people respond very well, some people not respond well. We still don't know what makes the responder versus non-responders. It's a huge area now. Immediately, of course, GWAS started. One paper came last year, another one came this year. Only gene they found was GLP-1 receptor itself. Okay, so it makes sense, right? That means that rest we don't know. So we still don't know what makes the difference. Nonetheless, then, so clinical observation know that effective GLP-1-induced weight loss is associated with a decrease in RER in humans, meaning that makes sense. It means active fatty acid oxidation. It makes sense because, again, I told you GLP-1 suppress food intake induces a fasted state. The peripheral tissue have to respond to that by start to use fatty acid. And then that is sort of the idea, maybe based on our work, maybe the carnitine synthesis may be important for that. So then to test this, we injected semaglutide into wild type mouse as a black, or no, sorry, the golden is a control, black is a knockout mouse. If in a major RER, if you inject semaglutide, RER drops in control mouse, makes sense because fatty acid oxidation is induced, but this effect is much less, sort of marginal in the knockout mouse. And then this is, I'm not going to show data, but if you inject semaglutide, serum isocarnitine goes up because fatty acid oxidation high, but this effect is not seen in knockout mouse, okay? Now, the key question is this pathway required for the optimum anti-obesity action of GLP-1? And to test this, we injected semaglutide GLP-1 receptor agonist into wild type mouse. It caused a substantial weight loss up to 5 days of injection. But this anti-obesity effect is much, much less in knockout mouse. When you give carnitine back, supplementation, you can actually restore this RER, and also the adipose tissue start to shrink. So then here, after 7 days of GLP-1, this adipose tissue morphology control lost fat. Knockout mouse did not really lose much fat, but if you add carnitine into this, then knockout mouse must start to lose it. So this is really interesting for us. Maybe what I told you so far is that the mitochondrial carnitine biosynthesis controls fuel switch into fatty acid, and it comes from this. And this is really required for not only cold adaptation, but fasting response and GLP-1 receptor analogs, the efficacy of GLP-1 on body weight loss. Now that this led to the question, maybe carnitine supplementation could be effective for those who need more carnitine. For example, vegans, strict vegetarians or vegans. And then really interesting, and then a couple months ago, my colleague Wash Yu contacted me. He's a clinician. Anecdotally, it's been known that carnitine supplementation is sort of alleviates the fatigue sort of feeling of after semaglutide treatment. And now they were in the planning to do this clinical trials. Okay. Do we have more minutes? 5 minutes? Okay. I want to end with speculation, okay, based on this speculation. Why do we eat meat? Oh, by the way, I love meat, and then love meat, and some people love meat, some people don't love meat, and then it comes to this, right? I'm really addicted to Sichuan food now. It says I'm really curious if anyone knows the best Sichuan food in town. I love to try Sichuan beef or anything. It's a spicy one. It's really, really interesting. I love to hear that. But this question is why you want to eat meat? And it's interesting. One of my daughters says, I don't really like the taste of meat. And I just don't get it. Why meat is so delicious. But if you think about this in evolution, it's more clear, right? Obvious. Carnivores eat more meat, and only eat meat, have to eat meat. Obligate carnivores have to eat meat, and herbivores don't need to do that. And evolutionally, if you have a lot of food around you, why you want to take risk to hunt something? It doesn't make sense to me. I mean, horse contain highest carnitine in their meat, but they're herbivores. That means that they have to make carnitine a lot. Because of this study, and because of the anecdotal conversation with my daughter, make me think: is it related to carnitine synthesis capacity? When I look evolution of carnivores and herbivores and across the species, herbivores, omnivores, insectivores, all kinds. And I asked some students or others, can we look the sequence homology of those sequences? And then, which is amazing, there are carnivore selective variants exist in this gene. There are 8 sites. It's just a little bit unusual. I mean, mitochondrial carriers are overall very, very conserved. But this is not only exist in the carnivores, okay, but not in herbivores, omnivores. Omnivore, carnivore, including humans, are very, very similar, almost identical. There are 8 sites. And some of them appear to change the angle of the helices or maybe potentially change the pore size. And then sort of, we can argue that. But this reminds me of what I told you earlier about UCP1, the evolution of endothermy, that which we've been studying for many years. And I told you UCP1 is a mitochondrial carrier. You associate 25 as 7. This molecule is thermogenic, only in mammals. Proton carrier. UCP1 gene exists in the fish, birds, platypus, marsupials, all kinds. But surprisingly, structure is similar, but they're not thermogenic. Okay, so this reminds me of this maybe variants during the evolution of mammalian species of this gene may affect the transport activity, and that could drive carnivore evolution. I don't know. Just like then Alice Ma, the Ph.D. student, BBS, and she's brave enough to test that idea. So she introduced carnivore selective variants in 8 sites into mammalian human form of SLC2545. We have now a reconstitution system, and then we introduced that into bacteria or mammalian cells, and see if this variant changed transport activity. So preliminarily, but it's amazing. She found that if you mutate those 8 sites to carnivore way, it blocks the TMO transport. Okay. So again, this really, it's still, the idea is that maybe there's variants occur in a carnivore evolution, limit the transport activity of TML, so that way they can't make enough carnitine, so that they may have to start eating meat. That's sort of the hypothesis perspective, but this is a testable hypothesis. So we're excited. Maybe the variants in this gene could be evolutionary driver of carnivore, like UCP1 evolution in endothermy. So with that conclusion and perspective, I'm really excited. Again, this is one example. I never thought about studying carnivore evolution, but identifying metabolism, unknown gene, and that led to the new question, and then unexpected observation. Now here we are. So I'm excited. We sort of start to deophanize some of the transporters. But the question key is like how those pathways are dysregulated in a metabolic disease, cardiovascular disease, aging, cancer, you name it. I think it's all open questions, and I think it's a great question. So with that, I want to end my talk by thanking people who did this work. This project was led by Chris Alger, helped with Hiroshi, Alice, and then Bo and Gilmore. They're from Collaborators Lab. And thanks to funding, and thank you so much for your attention.
Thank you so much, Shingo, for the wonderful talk. I will start with the first question. So I was curious, so you mentioned in carnivores, they will need to, so they eat the meat, and then that's where the carnitine is coming from. And then if they transition into vegan, and then mostly it's coming from the de novo synthesis pathway through here. And do you see, or is it testable to see whether the transporter expression get increased as well with this transition? And then also, is the trimethyl lysine, that is not primarily coming from meat, right? It's probably more like it's more like a
proteolysis.
Yeah. Proteolysis. Yes. Gotcha.
So the trimethyl lysine comes from muscle degradation, proteolysis.
I see.
And then I guess, of course, if you eat meat, it comes from trimethyl lysine exists. I think there are many theories out there, right? Of course, some of the cellulose digestion enzymes are lost and other things. But we're excited with the idea maybe the carnivore start to eat meat could be coupled with the deficiency of need or deficiency of carnitine in the body because of their body can't make enough carnitines. That's a one theory. In fact, the knockout mouse have the lower carnitine levels, and then we're always joking. Maybe we're going to make carnivore mice. They start to bite you more. But that is, we're now testing that idea as well. And then there are other enzymes that I described that is required for carnitine synthesis. Many of them are not really well characterized. Now we're knocking mouse to make sure that it's a sort of key enzymatic steps for the carnivore synthesis. One interesting observation now is that we have a way to adjust carnitine synthesis depends on availability. This can be seen in cultured hepatocytes. If you deplete carnitine in the media, hepatocytes make a lot of carnitine. If you add carnitine into the media, hepatocytes stop making it. How that really sensing, what is the sensor? How cells sense carnitine, and then how they stop that, what's the negative feedback loop? Completely open question. I'm excited with that. Yeah.
Have you tried to actually make the transporter specific knockout in the brain and see, does it make the mouse changing their behavior?
That'd be really cool. And that's it's interesting. This transporter is highly expressed in the brain too. And then I thought the neurons use glucose primarily, but some other people say that's not true. So it could be related to that too. Okay.
Hey, Shingo, nice talk. My question is more related to your broader topic about fuel selection and mitochondrial carriers. I'm just curious about, for so many carriers you have already studied, is there a more common framework or pipeline to discover their functions, or what's the differences in find?
I wish to know. I thought about, okay, we're gonna de-orphanize all of them, but for the last 7 years or so, probably we did 6 or so, right? It's much complicated than I initially thought. Initial idea was, okay, brown fat express large part of this carrier proteins. So we could systematically knock out or knock down and make mitochondrial liposomes. And then we add some mixture of metabolites to see what comes in and comes out. The idea, it did not really work. It worked to some extent. And for example, phosphoenolpyruvate worked out. It did not work for choline because there's other compensatory pathways. And so it's not really, it's more, so far what it worked out was almost like accidental, right? That means, for example, this gene there's a hint in the GWAS that I didn't show, but the GWAS data shows a clear correlation between circulating TML and this gene, right? So that was a good example. The phosphoenolpyruvate, mostly driven by the mitochondrial metabolite observations. So, and then 44 is mostly in the biology related to BCAA. So I wish to know some formula that works for all of them, but we don't.
Awesome. Thank you so much, Shingo.
Okay, thank you.
And then you have this certificate to honor your work. Okay, so now our final speaker of the session is Dr. Vamsi Mootha, professor of systems biology at Harvard Medical School and the investigator of the Howard Hughes Medical Institute, and a faculty member at Massachusetts General Hospital and the Broad Institute. Vamsi's laboratory has transformed the field of mitochondrial biology from defining the mitochondrial proteome and discovering key components of the mitochondrial calcium uniporter to uncovering the molecular basis of inherited mitochondrial disease. His talk today is titled Mitochondria at the Nexus of Genetics, Oxygen, and Disease. Please join me in welcoming Vamsi.
Thank you, Zin, for the kind introduction, and to all of you for inviting me to join you today. I really enjoyed Shingo's talk right now. I know that we already had a shared fondness for mitochondria, but I think we also have a shared fondness for spicy Chinese food, except I don't prefer the beef, though. I actually prefer my mapo tofu without any meat. And Shingo, I think Mountain House is actually a fairly decent place, so I don't know what others think, but I think Mountain House is actually fairly good. And Gourmet Dumpling used to be really, really good, but it's no longer in business. But I think there's better experts here to guide you. I'm really excited to share some of the science that we've been doing in our laboratory. And I just want to say that it's also awesome that you guys do this every year for such a long period of time also. There's nothing better than combining science with culture and have immense sort of admiration for classical Chinese culture. So awesome that all of you are continuing this tradition for so long. So I'm going to dive straight in. And so without doubt, one of the most important events in the history of complex life on our planet was the acquisition of mitochondria through this process called endosymbiosis. And to the best of our knowledge, this was a singular event. All eukaryotes on our planet can be traced back to a mitochondrion containing last eukaryotic common ancestor. And this is distinct from the chloroplast. That's actually been acquired at least 2 independent times in evolution. So this organelle really is unique. And then over a more than 1.5 billion year period, that original bacterial genome has become tinier, relegating most of its coding capacity to the nuclear genome, so that both of these genomes now have to work together, both in health and in disease. I've long been fascinated by mitochondria. I was actually a medical student working here in Judah Folkman's supergroup some 33 years ago. So I only worked in that group for about 1 semester. I really didn't enjoy that type of cell culture work. It didn't suit me well. But I do have fond memories of being right over here in the Longwood Medical Area a long time ago. But soon thereafter, I actually developed a passion for mitochondria. And as all of you know, this organelle is implicated in a spectrum of diseases. As we all age, the activity of mitochondria goes down. And the billion-dollar question in the aging field is cause and effect. Is this actually driving the aging process, or do you just have sick tissue, so you have sick mitochondria? And then at the opposite end of the extreme are a very large number of ultra-rare diseases, including diseases such as Lee syndrome. These are typically monogenic disorders where a child is born with a birth defect in the mitochondrion. And there's no doubt that the mitochondrial dysfunction drives the pathology. And so as I was finishing, I had developed a passion for this organelle in the early 90s, and that's what my medical school thesis was focused on. That's what I focused on as a clinical house officer over at the Brigham. And I was actually graduating from the Brigham in the year 2001. And at that time, all of the focus on rare mitochondrial diseases was on the mtDNA. For those of you that are clinically trained, you know that if ever there's a board's exam question about mitochondria, the answer is always maternal inheritance. But the mtDNA only encodes 13 proteins total. And 2001 is also the year that the human genome was coming online. I had a background in math and computer science, and so I wanted to try to combine that background with the new tools of genomics to try to understand what is it that the nuclear genome is doing to build mitochondria. And the reason we knew so much about that tiny little mtDNA is because Fred Sanger and colleagues had used first generation DNA sequencing technology. And one of the first whole genomes to be sequenced was actually the human mitochondrial DNA from a collection of placentas in Cambridge, UK. And then from that, we've long known about the coding capacity of the mitochondrial genome. But then 20 years later, this other genome was coming online. And I trained both with Matthias Mann to learn mass spec proteomics in Denmark. Then I came back and trained at the Whitehead Institute with Eric Lander when the human genome sequence was just coming online. I worked on the mouse genome annotation team. And after I set up my own laboratory over a more than 15-year period, what we've really focused on is trying to build a protein parts list for this organelle. We've combined protein mass spectrometry. We've developed very simple machine learning algorithms, as well as large-scale epitope tagging studies to basically construct a parts list for this organelle. And we're up to Mitochondria 3.0 now, an inventory of about 1,100 proteins made by the nuclear genome that comprises organelle. We now have pathway annotations, we have suborganelle localization. It's freely available. And the cool thing is 99% of this organelle is encoded by this nuclear genome. And so with that as an introduction, I'm going to share with you kind of two kind of arcs from our lab. And the first one, I'm going to share with you some very recent work that addresses the question of how do these two genomes get coordinated? And then in the second, I'm going to talk about some of our approaches to try to alleviate the disorders of the mitochondrion. And then at the very end, in one slide, I will make an attempt to combine these. Okay, so how do these two sets of genomes get coordinated? And this is no mean task because in contrast to the nuclear genome, the mtDNA, it's fast evolving. It's about 100 times more rapidly evolving. It's maternally transmitted in contrast to the nuclear genome, which is diploid. This is high copy number genome. And the nuclear genome is diploid, so you either have homozygotes or heterozygotes, but the mtDNA, some cells have thousands of copies of mtDNA, so you can have a mixture of, quote, wild type and mutant molecules. So this creates all sorts of interesting dynamics. And importantly, all of the factors, the protein factors you need to replicate the mtDNA are nuclear encoded. So clearly, there's got to be some crosstalk. How are we going to study this? This is very, very, very hard to study. And this is something that I've long been interested in trying to investigate. And we've been using a biobank scale approach. So I think all of you are familiar with things like UK Biobank and all of us. These are these vast databases where you have patient phenotypes. You also have genome sequence as well, and they're freely available. Now, most of these analytical pipelines will discard the centromere, the telomere, the X, the Y, and the mitochondrial DNA. And so Rahul Gupta, a former MD Ph.D. student, now an intern at, or now a resident at Mass General Hospital, he was jointly mentored by myself and by Ben Neal. And basically what we did is we went dumpster diving. We took all of the reads that did not get mapped to the autosomes, and then we reconstructed for each of 750,000 people, each person's self mtDNA, that's a self-reference mtDNA. So that's one thing that we computed. From the relative read coverage, we computed the mitochondrial DNA copy number for each person. So that's a trait, okay? Just like body weight or height, that's a trait. And then finally, for each heteroplasmic variant, we also determine what the heteroplasmic fraction is. So one self-reference DNA and then two traits or two sets of traits. And then you can do a lot of cool stuff. I'll just share with you a few of the things that we learned using this approach. So the first thing is mitochondrial DNA copy number declines as a function of age. And we did a lot of work to correct for variation in blood cell composition. This is an extremely strong marker of the aging process. In the same way that you can do GWAS for height or for body mass index, you can also do GWAS for mitochondrial DNA copy number. And in this Manhattan plot, you get a lot of loci, meaning that variation in these loci influence variation in the abundance of mtDNA copy number. And if you knew nothing about the nuclear factors required to replicate mtDNA, you would basically rediscover virtually all of those through this Manhattan plot. Everything shown in red is a mitochondria protein encoding locus, and we have all the replication machinery that's nuclear encoded. The other half of these hits are basically uncharacterized as of right now, so lots of opportunity to discover new mechanisms by which mtDNA copy number is controlled. Now, one of the things that I've long been interested in is mtDNA mutations accrue as a function of age. This is one of the strongest biomarkers of the aging process. And from the UK Biobank, we also see right around age 60, this hockey stick moment where mtDNA mutations are now detectable in the blood. And we're very curious to know what the mechanism is. Okay? Now, for decades, this is what's been kind of in the textbooks. It's this free radical theory of aging where you have normal mitochondria, but they're always producing some reactive oxygen species. Those reactive oxygen species damage the mitochondrial DNA. That damaged mitochondrial DNA encodes other components of the electron transport chain. So now they generate more ROS. You get this vicious cycle that spirals out of control, and you get this aging problem. So that's the null model. Now, from 750,000 people, we can actually look at all of the classes of mutations that either do or don't accumulate as a function of age. And what we immediately see is that there's this A to G and C to T pattern where it's not all somatic mutations or SNVs that are accumulating. It's only certain classes. And there's a very, very stark strand asymmetry as well. And the mitochondrial aficionados know what this is, but I'll just tell you up front, this is not an oxidative stress signature. These are transitions. These are not transversions. This is not what you typically see with free radical damage. What the mitochondrial aficionados will know is that this is the mutational spectrum for the polymerase. Okay? These are simply replication errors. When mitochondrial DNA replicates, it replicates via strand displacement model. One of the strands is actually kind of naked for much of the replication cycle, and it's prone to deamination. And so it's known from quite a bit of decades of molecular biology research. That's what this is. So then, if these are replication errors, is there any evidence of selection? And so what we find, just focus on this rightmost column over here, what we find is that loss of function variants, missense variants, as well as synonymous variants, they all appear to be accumulating. If there was strong selection, either for or against, we wouldn't have gotten this pattern. Then we can ask, what is the distribution of heteroplasmy for the age accruing heteroplasmic variants versus the ones that are not age accruing? And so what's shown over here are all of the variants. We basically partition those into two disjoint sets, either those that accumulate or those that don't. Now for those two sets, then we can ask, what does the heteroplasmy distribution look like? And I hope you can appreciate the class that's accumulating tends to be a much lower fractional heteroplasmy. Most mtDNA variants are functionally recessive. Okay? And so these appear to be just low levels of errors. Right? They're so low, they're typically not going to have an impact on the mitochondrial bioenergetics. And so what we're finding is that the variants that are accruing appear to be neutral, right? And we don't see any strong signs of selection. So if there's no selection, then why are they accumulating so sharply at the age of 60? So we didn't know, but in the same way we didn't know what controls mtDNA copy number, we asked, why don't we use the burden of mtDNA heteroplasmy as a trait? With 750,000 people, let's do GWAS and see if it teaches us the mechanism. When we did that, we can do a GWAS, we can do an RVAS, we get Manhattan plots, but nothing is painted in red here because nothing here is related to mitochondria. Okay? So these are all nuclear, of course, they're all nuclear genetic loci, but none of these actually encode mitoproteins. Okay? And so we get things like TERT, we get things like TCL1A. If you do a quick Google on these types of things or PubMed, these are all either inherited variants or somatic variants that have been associated with something called clonal hematopoiesis. This is a type of somatic mosaicism. And as all of us age, you can just get certain clones that kind of outcompete other clones. And Ben Ebert here in Boston termed this clonal hematopoiesis of indeterminate potential. And so the same genes that they identify for that process are the genes that we identify over here. Okay? So this is our model for what's happening. So you just have a bunch of cells, okay? And the mitochondrial DNA is just replicating, and it just has a higher mutation rate, 10 to 100 times higher. And so you're just all, as perfect as you think you may be, you have mtDNA mutations at low levels. Okay? But these are really not functionally relevant. Now, once a cellular clone takes off for whatever reason, okay, that's going to bring along with it any mtDNA variants that have been marked. And so those are just coming along as passengers. Okay? So that's our model for what's happening. Lots of people have proposed that high mtDNA mutation rates are associated with a lot of diseases. We're only focusing on blood. Most diseases do not appear to be associated with this high MTSNV count. However, the bright red all correspond to blood cell dyscrasias. So it does appear to be the case that high mtDNA mutation count is a marker for future blood cell dyscrasias. This is consistent with the idea that CHIP is a precancerous lesion. And there's a lot of excitement about this concept of somatic mosaicism because this may extend to other tissues as well. And we actually think that the mtDNA mutation burden may actually serve as a more sensitive marker of somatic mosaicism. So that was our original GWAS. Sort of the gurus of the CHIP world are able to call all the samples in UK Biobank that have CHIP mutations. There's about 17K. We can take those away. If you take those away with the remaining 483,000 samples from UK Biobank that are not known to have CHIP, if you do GWAS again, you actually get the exact same Manhattan plot, meaning that those are the germline variants that are predisposing these individuals to get CHIP, even though it's not detectable by traditional methods. So that's sort of our first story. It's kind of exciting because it seems to be the case that this mtDNA mutation burden signature, telomerase, as well as clonal hematopoiesis, they actually may all be related to each other. It's all kind of one process, basically. That underlying process appears to be somatic mosaicism, but why that's going up with age is still an open question. Okay, now I'm going to shift gears completely and talk about a very different topic, and that is, can we develop medicines for mitochondrial disease? And I want to just introduce these rare mitodisorders that we've been focused on for a long time. I'm typically talking about a defect of the electron transport chain or the OXPHOS system. At this point in this talk, hopefully you appreciate that they can be due to either mtDNA mutations or nuclear DNA mutations. There's a lot of phenotypic heterogeneity as shown in this slide. And we have very limited therapies. There's actually now, believe it or not, 3 FDA-approved medicines for 3 rare mitodiseases, but they barely work. And there's more than 300 monogenic forms of mitodisease. Of the 300 monogenic forms of mitochondrial disease, a few of them are common, meaning there's at least 1,000 cases in the United States you can identify. And that's an important number because if you want a biotech company or a pharma company that you develop a drug, you really need to have at least 1,000 patients that you can point to before they'll do so. But most of these disorders are, they're called N of 1, but you actually can't publish in Nature Genetics with only 1 case. So usually they're N of 10 diseases or N of 100 diseases. But these are ultra rare diseases. And for a long time, we've been interested in trying to find therapies that might suppress diverse forms of mitochondrial dysfunction. And so what I want to do is I want to share with you an arc that's been developing in our lab over an almost 10-year period that began when Isha Jain was a graduate student, and she's now leading her own independent group at UCSF. Slil Ast continued this work and extended it into the most common monogenic mitochondrial disease. She's now leading her own group at the Weizmann. Josh Mizell was joint between my lab and Gary Ruvkin's laboratory, really pushed us, what I'm about to tell you, into the worm model. He's now a professor at Brandeis. And Rob Rogers is a talented former chief resident and hardcore physiologist that's now at a Tim Springer company called Tectonic. I'm going to share with you some of their work over the years. So 10 years ago, right when CRISPR technology became available, we received from Feng Zhang a genome-wide CRISPR library, and we did the following screen. We basically CRISPR mutagenized cells, And then we added different types of mitopoisons. And when you add mitopoisons to cells in culture, you get a proliferative defect. And we were looking for a suppressor that would suppress the mitochondrial toxicity. And when we looked at about 60,000 guide RNAs, like number 1, 2, 3, 12, and 14 guide RNAs all corresponded to the same gene, and that gene was von Hippel-Lindau. And as all of you know, von Hippel-Lindau, VHL, is a gene that's important in the body's oxygen sensing pathway. And what the gene taught us, what the screen taught us, is that when you have mitodysfunction, genetically tricking cells into thinking that they're hypoxic might actually be beneficial. And this actually may seem a little bit paradoxical, because even at that time, there's some doctors that were saying, hey, your kid has mito disease, give them more oxygen. Now, if you just think about that for about 5 microseconds, you realize that that's illogical, right? The mitochondrion consumes oxygen. So if it's broken, if anything, you have too much oxygen. So it's illogical to actually give more oxygen. And in fact, the screen actually supported that more logical explanation that maybe low O2 would be beneficial. And so we had a mouse model of Lee syndrome. This is a mouse model of complex 1 deficiency that was developed by Richard Palmiter's group. And these mice are born okay, but they usually die at about 2 months of age from their neurological disease called Lee syndrome. So we had these mice, we had this CRISPR result, and Warren Zapal he passed away a couple of years ago, but he was my friend and colleague at Mass General Hospital. He studies pulmonary hypertension. He had a protocol already in place for placing mice at 11% oxygen. So that's sort of the equivalent of 15,000 feet elevation. So healthy humans should be able to tolerate it. So it's not Mount Everest, but it's right closer to base camp. And so what we did is, after weaning, we placed right around day 20 or 25, we actually placed 2 of these mice in hypoxia and see if you can guess which 2 those are. Hopefully it's super obvious. We actually thought that there were genotyping errors, but there are no genotyping errors. These mice ordinarily die at about 60 days from their neurological disease. But if they're breathing 11%, so the equivalent of living at 15,000 feet elevation, this is a survival curve. We carried this out post-publication. They survived about 300 days of age. Conversely, if you dial up the oxygen, they actually die within about a week or so. And this is actually consistent with what we had heard anecdotally from patients. After we published this paper in 2016, I received phone calls from multiple clinicians who shared with me anecdotes of their mito patients, outpatients that were placed in hyperbaric oxygen, that actually 2 of them actually died within 24 hours of HBOT therapy. 2 of them actually became blind within 1 week of hyperbaric oxygen. So those are anecdotes, but I think those anecdotes in combination with our mouse data strongly suggest that supplemental oxygen, when it's not needed in a patient with ETC deficiency, may actually be a bad combination. Subsequently, we asked whether hypoxia would reverse any of this pathology. So these mice exhibit a very stereotyped trajectory. And so living in normoxia, they will hit euthanasia criteria at MGH right around close to day 60 or so. So we started hypoxia then. They regained body weight. We can actually make these T2 intense lesions, which are reflective of a lot of neuroinflammation. We can actually make those disappear after about 3 weeks of hypoxia therapy. And so we're very excited by this initial result, because for a long time, we've been looking for some sort of a suppressor of mitochondrial disease. And it seemed that we had found an environmental perturbation, this low O2, that was powerfully suppressing. And conversely, high O2 was actually exacerbating. So it's really cool that we found something that we could push to the left or to the right and have such a profound effect. And since then, we've been working on the mechanism, the generalizability, and how we're going to translate this into the clinic. When you get this type of a discovery in your lab, on the one hand, you're super excited and you want to move a lot of your lab into that direction. At the same time, you want to make sure that this is not some real weird thing that's private to the NF-S4 knockout mouse. There's only 1 patient in North America that's NF-S4 knockout. And will this even generalize to that person, or is this just a funky mouse phenomenon? over the last few years, we've been able to show that other mouse models of mitochondrial neurological disease are also potently alleviated by low O2, including Friedreich's ataxia and Parkinson's disease due to synucleinopathy. These brains typically have a secondary mitochondrial complex 1 deficiency. And when we start hypoxia after the synuclein injection, these mice actually show motor deficits. And then once we start the hypoxia, these motor deficits actually reverse. It's actually very exciting. They've lost neurons at that point. The neurons that are lost are not coming back. Okay? It's just that there are still some neurons there at this point. And once you put them in low O2, the motor phenotypes are much improved. Okay? So what's been exciting is that hypoxia appears to be extending to multiple forms of brain disease for reasons that we do not know. It is not helping cardiac disease in these models. So in the Friedreich's ataxia model, for example, this actually continues to die from its cardiac disease, and we don't know why, but its ataxia phenotypes are improved. We've also done low-high oxygen CRISPR screens. We've identified more than 200 genes. If you knock them out, you'd call them like essential genes in normoxia, but under low O2, they're okay. So these are almost like temperature-sensitive mutants, but we call these OxyS mutants, oxygen-sensitive mutants. And this helps us to sort of nominate candidate diseases that might benefit from hypoxia therapy. I like to tell my lab what happens in cell culture stays in cell culture. I can rescue almost any mito defect in the laboratory. I can even grow cells in the laboratory lacking mtDNA. I can guarantee you that you cannot grow an entire mouse without mtDNA. And so while these cell culture things are awesome, it's not sufficient. And one of the models that we've been very excited about over the last couple of years, it's intermediate between cell culture and mouse, and that's the worm model. So Josh Mizell, who's a joint between my lab and Gary Ruvkin's laboratory, has been able to show that the very models that we're able to rescue in mice, we can also rescue in worms. So if you don't believe this because of the mouse work, hopefully you're now convinced either because you like worms or because you like evolution. And so in worms also, some of these wild type worms are fine under high O2, but these complex 1 mutants just get fried under high O2. So these are also oxygen sensitive. And what's really interesting is for the longest time, no one's ever been able to make a complete knockout of frataxin. Okay? Frataxin is the gene that underlies Friedreich's ataxia. It's really important for iron sulfur clusters. If you try to knock it out in a worm, they all die. Now, if you knock it out under low O2 conditions, not only do the worms survive, but they actually complete their entire reproductive life cycle. And so that's been very powerful. I'm not going to go into it, but that oxygen sensitivity has allowed us to now perform genetic suppressor screens, secondary suppressor screens, to work at some of the mechanisms by which hypoxia is alleviating these mutations. I'll share with you what we've learned at a high level in terms of what are the mechanisms of oxygen sensitivity. So imagine that you have an enzyme, it could be a metalloenzyme that catalyzes A to B. This may have actually evolved under anaerobic conditions early in our planet's history. Now, as soon as oxygen appears on the planet, oxygen can now react with a few things. It can't react with everything, otherwise we would spontaneously combust, but it can react with ferrous iron. And when that happens, this metalloenzyme is now damaged. When that electron falls into oxygen, you get a partially reduced oxygen molecule. And that's what a lot of people have focused on historically, but you also get a damaged enzyme. Okay? So we're identifying different sort of complementation groups that are oxygen sensitive. So there are some proteins that appear to serve as physical shields that literally block the access of oxygen to a metal center. You can imagine that protein is only essential when you have high O2. Respiration also helps to eliminate this oxygen. So if you can't respire, now you become more vulnerable to high O2 because you can't suck away the O2. Similarly, you might have some ROS scavenging enzymes. Those are going to be important here, but if you don't have any ROS, you can probably dispense with those. And so those are some of the mutational classes, mechanistic classes into which our oxyS mutants fall. And I think this paradigm also helps explain the difference between hypoxia therapy and antioxidants. I put up this slide because I always get annoyed after my talk if people start asking me about ROS. And I want to make it very clear, hypoxia therapy is totally different than antioxidant therapy. Antioxidant therapy does not work in any of the models that I've shown you. They have failed Lee syndrome trials. They have failed Friedreich's ataxia trials. I think what is happening is in this reaction that I just showed you, there's two halves to a redox reaction. Traditional antioxidants, catalase overexpression and superoxide dismutase overexpression, they deal with one half of the reaction. But the other half of the reaction, you can only kind of prevent by working more upstream. So we think hypoxia therapy may be operating more upstream. I'm showing iron simply as an example, but many, many classes of molecules can get oxidized by oxygen. And so this has led me to speculate. I've seen patients with mito disease for decades, and one of the interesting observations I've made is you never find complete knockouts of core components of the electron transport chain. They're probably just not viable. Right? So what I think is happening is, you have a wild type fetus that's going to be fine in utero. It's going to grow up. It's going to be fine. If you have a mutation in an essential core gene, okay, that's going to be a fetal lethal. But then there may be another class of mutations, okay, oxy-S mutations, that are actually tolerated in utero. The in utero environment is actually hypoxic. So is it possible that the hypoxic in utero environment actually shapes the allelic spectrum of mitochondrial disease? So that these are actually buffered here, but then once these kids are born, it's like a four-legged stool that's now a three-legged stool. They're vulnerable. And now when there's another insult, they actually have a catastrophe. So that's a hypothesis right now. I'd love to think of a clever way of actually testing this hypothesis. It's not easy, but I just wonder if that's what's happening. Okay, in the final few slides, I've shared with you some of our work on hypoxia as a medicine, but how do we harness this truly as a therapy? Everything that I've shown you so far requires chronic continuous hypoxia. It's like moving to Tibet, which could be fun, or it's like living in a bubble, which may not be fun. Okay? We've tried more practical things. Antioxidants don't work. HIF activators are not sufficient in the context of a whole organism. Intermittent regimens don't work. In fact, they can even be bad because during the hypoxic phase, you get an erythropoietic response, then you go into normoxia. It's a gush of hyperoxia. So we need chronic continuous hypoxia. So how are we going to make this into a medicine? And so one possibility is to repurpose what the sporting industry has already invented. There's a company called Hypoxico that's in New York that actually makes tents and face masks and nitrogen generators that allow athletes to train at high altitude. Okay? So we actually completed a phase 1 safety tolerability study at Mass General Hospital on the 12th floor at MGH. And this was conducted by Lorenzo Berra, one of my colleagues. And so we actually brought in 5 healthy volunteers, and over the course of about 5 or 6 days, brought down the oxygen to about 11%, showed that we could actually deliver it, at least on a short-term basis, in a safe way without any major adverse events. And so it's conceivable that in the same way people walk around with tankless oxygen, one day people might walk around with tankless nitrogen as well to dilute the air that they're breathing. So that's a possibility. But of course, what would be very, very convenient is if we could actually bottle this up into a pill. And we introduced the concept of hypoxia in a pill a few years ago. And our vision was to take 2 drugs that basically hematologists have been developing for completely different purposes. There's a class of molecules called the GBT series of molecules. These are hemoglobin affinity enhancers. So Linus Pauling had showed in the mid-1940s that when hemoglobin is oxygen bound in that tight conformation, it tends not to sickle. And so this company, Global Blood Therapeutics, developed small molecules that bind to hemoglobin and park it into that tight conformation. So they like the drug because it tends not to sickle. We like the drug because it tends not to offload its oxygen. It's a left-shifted hemoglobin. And so that's why we like this drug. It causes tissue hypoxia. The problem with using that as monotherapy is physiology kicks in. If you go to altitude, you're going to get an erythropoietic response, and now that's going to try to defend oxygenation. So to block that, there's a drug that Steve McKnight's company Peloton had developed. It's a HIF2 alpha inhibitor for VHL-driven tumors, and that will actually blunt the erythropoietic response. And so when we use both of these drugs, hope you can appreciate that in that first model, these mice, we take them until they have advanced disease. So this is a very, very stringent test, and they'll die in the next 10 days from their neurological disease if they get vehicle. If they're getting this combination, now they're surviving median in excess of 160 days. Both of these drugs have been shown to be safe in healthy humans. And so we're excited about this as a potentially pharmacologically tractable form of hypoxia in a pill. So I'm going to end now and just say that when we think about human disease, we think about genes and environment. And I think when it comes to mitochondrial genetics, I think the key environmental variable might actually be ambient oxygen. And the reason is we spoke earlier about this endosymbiotic event that took place about 1.8 billion years ago, but that was following another important event on our planet's history, the Great Oxygenation Event. That happened, so these bacteria were already learning how to cope with high oxygen. They were evolving mechanisms, including proteins like frataxin, including proteins like NDFS4. Those were actually acquired later, probably, following the Great Oxygenation Event. And those actually allowed these bacteria to specialize and function, even in the presence of high O2. And now I think what happens is today, a lot of the mito diseases can be understood in the context of these two great events. A lot of these mito disorders can be Mendelian or maternally transmitted. And a lot of these mito disorders, if they have mutations in some of these specific proteins, they'll exhibit oxygen-sensitive phenotypes. And so it's always fun to try to sort of contextualize your work in sort of evolution. And we think that both the Earth's history as well as what happened with complex life may be very relevant for our understanding of mito diseases today. So with that, I have acknowledged all of the amazing current and past members of my laboratory. I want to acknowledge my collaborators as well as the folks that provided these mouse models. And I want to thank you for the invitation to come and join you today.
It's awesome talk, Vamsi. So questions?
Okay, thank you, Professor. Very impressive talk. I would ask us maybe some naive question. When you talk about the 11% oxygen, what's the other gas? Where the effect comes from the other gas?
Yeah, we're diluting the air with nitrogen.
I see. Thank you so much. That's what
So when we talk about the oxygen, could that be different cells have different demand of oxygen? So for the general cell, for the 21% oxygen may be enough. But for example, for the highly active neuron, maybe they are so demanding, so this same person oxygen become hypoxia to them.
Yeah, I actually think each tissue is probably specialized during development to sort of take advantage of and cope with, whatever its O2 tensions are. What I'll note is that when we do cell culture, we're always using 21% oxygen. There's not a single cell in your body that actually sees, maybe in your buccal epithelium, but otherwise even in your lungs, because of gas exchange and humidity, you only see 15% O2. And so a lot of what we do in cell culture is kind of artifactual. Every single tissue is seeing different O2 levels. Sometimes it's tightly controlled and sometimes it's dynamically regulated. And I think we just need better methods with which to sort of measure and manipulate oxygen so that we can try to understand these things. One of the things that we've observed is that hypoxia therapy is very good for the brain. It's not helping the cardiac disease, and we don't know why. And it may be because of differences in oxygen handling, or it may be because of other differences in metabolism. We don't know.
Great talk as always. So the CRISPR screen identify VHL, and then you mentioned that it not really work in vivo, right? And then is that because of this sort of consequence of VHL deficiency, or could you tell us more about that?
Yeah, no, we actually tried knocking out Ph.D. 1, 2, Nestin Cree Ph.D. 3. We got the Chuvash mutation for VHL, cross them to the NFS4. Not only do they not work, Shingo, a lot of them even make things worse. And the ones that make them worse also bring up the hematocrit as well. And so in cell culture, it's a fact that knocking out VHL helps. It jacks up glycolysis. It actually helps in cell culture. So hence, what happens in cell culture stays in cell culture, because in vivo, when you do that, you get a greater red blood cell mass, and now you get tissue hyperoxia as well. We can actually prove that because in those instances, we make things worse. If we block that with the HIF2 alpha inhibitor, we now remove that worsening.
Crazy idea is that a carbon monoxide, or anyone actually do that? Is that a too dangerous?
No, let me see. We actually did
oh,
that.
okay.
Yeah. Let me see if I can find it quickly. Otherwise, we've done carbon monoxide. No,
it seems like easier to apply if you low dose of carbon monoxide.
Yeah, it actually sounds crazy, but it's actually totally right. And so we actually, in a 2019 paper, we actually showed that low dose carbon monoxide. You see this now? So if you take these mice to the brink of death, and then you start carbon monoxide, you can actually reverse this neurological disease in this model also. So sublethal doses, they will die a few weeks later from the lesions that are classic for carbon monoxide. But in a lot of ways, this is a lot like the hemoglobin left shifter. So carbon monoxide finds, parks it in that tight conformation. And in fact, Linus Pauling even proposed carbon monoxide as a therapy for sickle cell as well. And so the beauty of this is the mouse actually clears its carboxyhemoglobin very rapidly, but humans don't. So we could almost imagine puff therapy, where every morning you take a puff of CO, it lasts longer. But that's a narrow therapeutic index, and I think there's enough kind of the PR around carbon monoxide is bad enough so that it's going to be pretty hard to advance. But in principle, that could work also.
Yeah. So you showed two drugs in one of your slides. One is oxybrother. That one I know little about, but the other one is like belzutifan. That is usually widely used for kidney cancer. And as far as I know, it's just toxic. When the patient is on it, it's going to have anemia, that kind of thing. So I just wonder if any of the drugs you're going to use, is going to be like specifically deliver, like a target deliver to the organs affected. Let's say if the brain is the organ that causing problems, so probably just do like a intracranial delivery that helps other organs not get toxic effect.
So first of all, Shingo asked a question about carbon monoxide. I'll just point out that in that same paper, we also used severe anemia in a mouse, just as proof of concept. That also helps to rescue this model because, of course, if you have anemia, you have less O2 carrying capacity. So that also helps in this mouse model, FYI. And the HIF2 alpha inhibitors, they also cause anemia. What we've shown is that the hemoglobin left shifters, they cause a polycythemia. Now, when you have the HIF2 alpha inhibitor, it actually brings it back down to normal levels. So the hematocrit and the hemoglobin go up with the left shifter, and that gets blocked with the HIF2 alpha inhibitor. So they're actually back to kind of normal levels. But you're right. I mean, these can be toxic drugs also. It's something we have to be very, very careful about.